Okay, so we are back here today to talk about the bony fish. So we talked about the cartilaginous fish in the last lecture when we talked about class Agnatha and also again class Chondrichthyes. Both of those being that they have cartilaginous skeletons. Class Agnatha were the jawless fish, and class Chondrichthyes were the jawed fish that had the cartilaginous skeleton. So now we're going to get into class Osteichthyes. And you already kind of know some of this. There's going to be overlap because we talked about, the Osteichthyes when we were going through class Chondrichthyes because it was easy to make comparisons between the two. So hopefully this lecture won't be too difficult and that you've already kind of had an introduction to some of these. Really quick, for your fish quiz, anybody know what fish family this fish belongs to? Hint, look on the caudal peduncle. There's a discoloration there on the caudal peduncle to let you know that there is a sharp spine here, sharp like the scalpel of a surgeon. So it's surgeonfish. So anyway, I just thought I would throw that out there. But again, these are going to be all bony fish. So again, bony fish are in the class Osteichthyes. And they have these skeletons, again, that are made of bone. And about 96% of all fish are going to be your bony fish. So the majority of your fish, that's why, again, when you're out there diving, you're going to see mostly bony fish. Again, you'll get to see sometimes the occasional cartilaginous fish. You'll get to see a shark or a skate or a ray. But again, the majority of the fish that you're going to see out there are going to be your bony fish. So again, they make up about half of all vertebrates because, again, about half of all vertebrates are fish, and the majority of the fish are bony fish. So bony fish, again, are both marine and freshwater. So when we look at bony fish, and they have different scales than cartilaginous fish because their scales here are going to be made of bone with this kind of overlapping mucous membrane. So they have these cycloid or ctenoid scales, and the scales are thin, flexible, and overlapping. And again, these are bony scales. The gill covering that they're going to have so the big difference here between cartilaginous fish is remember that cartilaginous fish are going to have anywhere from 5 to 7 gill slits. Here in bony fish, there are no gill slits because they have this big gill chamber where they have all of the gills in one gill chamber. Instead of, in your cartilaginous fish, you've got anywhere from 5 to 7 gill chambers with one set of gills in each gill chamber. Here in bony fish, all of your gills are in the same gill chamber, and that gill chamber is covered with what's called an operculum. That's your gill cover. So remember that operculum is that term that we used when we talked about mollusks, and especially gastropods or snails. And remember that when they would pull that muscular foot in, at the end of it, they had an operculum that would seal off that opening called the aperture. So again, typically we think of an operculum as something that's kind of covering an opening. So in this case, our operculum covers the gill chamber, okay? So that gill covering, again, is called an operculum. It's spelled the same way. So hopefully maybe that'll help you remember it. Again, these guys are going to have a homocircle caudal fin. So remember, let me just go to a picture of a fish here. Remember that homocircle caudal fin just means that the upper lobe and the lower lobe are going to be equal in size. This is different than your cartilaginous fish, again, that had the heterocircle caudal fin, as you remember. Okay, so again, these guys are going to have these highly maneuverable fins. And that's because their fins are not stiff like the cartilaginous fins are. Okay, and they're also retractable, so they can pull these fins up and down. So these fins are going to consist of these thin membranes that are then supported by these bony spines that we call the fin rays in between. So again, let me show you this on this fish here. So again, you'll see these bony spines sticking up. Again, some are larger, and we call those hard spinous. And then some of these bony spines are really thin, and that makes this ray soft spined. So again, they're still going to have these bony spines sticking up and then this thin membrane of tissue in between those spines. So that's what's going to make up your fins. And again, we have these fin rays, and we typically will use the term with these fin rays. Let me go back here. Hard spined. Again, so this is a hard spined fin ray or hard spinous, because it has these kind of thicker bony spines. And then here with a thinner bony spines, we call this soft ray. Okay, so hard spine and soft, hard spinous and soft ray. So then we also are going to see in the bony fish a terminal mouth, which means that it's just kind of located at the very anterior end of the fish. Remember that in your cartilaginous fish, they had this very ventral mouth. So again, I'm just going back and forth here. So here's that very terminal mouth we call that terminal because it's at the very end of the anterior end of the fish. It's not dorsal and it's not ventral. It's just at the very tip, the terminal mouth. So, and again, that is different from your cartilaginous fish, which have a ventral mouth. The teeth are going to be attached to a jawbone here, okay? So a little bit different than your sharks, where their teeth were just kind of loosely embedded in that membrane around the jaw. So here the teeth are actually attached to the jawbone, and the jaws here are going to have more movement than sharks. So they're going to have these protrusible jaws, especially the lower jaw can protrude forward. So again, a little bit different than your cartilaginous fish. And then here, big difference is going to be how they deal with buoyancy. And I'll talk about this and show you some animations of this. But bony fish are going to use a swim bladder. And this is a big gas-filled sac that's just above the stomach and intestines. And they're going to use this gas-filled sac to control their buoyancy. So I'll show you an animation of how they do this, kind of using Boyle's law here in just a little bit. So again, that swim bladder is how they're going to control their buoyancy. Now remember, sharks don't have that swim bladder, so they had to rely for buoyancy on a really oily liver. They had to rely on these, stiff, very triangular pectoral fins that act kind of like wings to provide some lift. And then they also had to rely on that heterocircled tail to help push the head up as they swim. So again, very different, big differences here between your class Osteichthyes and class Chondrichthyes. And when you look at your bony fish, obviously, because there are so many fish in this class, they're highly varied in shape. And of course, their shape is going to fit their function. So their shape is going to fit their feeding niches and the habitats that they live in. And we'll talk about that. Okay, so remember these guys have these cycloid or tenoid scales. The cycloid scales will typically be smooth, while tenoid scales have these tiny little spines along their exposed border. And I'll try to remember to put a cycloid scale, a tenoid scale, and a placoid scale underneath the microscopes for you this week in lab. Again, here difference is that the cycloid and tenoid scales are made of bone, and then they're covered by a thin layer of skin and a protective mucus, okay? And that mucus is to help protect the fish from parasites. And those of you that have had aquarium fish know that this mucus coating is very important for them. So if the fish is stressed and not producing that mucus coating, it makes them very vulnerable to parasitic infection. So that's why typically when you move fish from, one aquarium to another, and maybe, you don't quite do it the right way, and the fish is quite stressed because it had a rapid change in temperature and things like that. then the fish becomes stressed and it doesn't produce that mucus coating. So when it doesn't produce as much of that mucus, it becomes vulnerable to parasitic infection. So anyway, again, just a couple things to remind you of bony fish characteristics. Again, terminal mouth that has no operculum gill cover. Again, movable pectoral fins. Okay, big difference there. Pelvic and anal fin, again, also retractable and movable. You'll see here they have the cycloid or tenoid scales, the homocircle tail, and again, the dorsal fins with fin rays, again, that can be retracted. So again, those are some of your major differences. You can see all these major differences. It's a nice review of the differences between cartilaginous fish and your bony fish here. Bony fish anatomy. So again, just as a reminder of what your fins are, hopefully you know these pretty well by now because you're going to use these to help you identify your fish-to-fish family. So again, we have our dorsal fin here. We have our caudal fin here. In this case, it's a homocircle caudal fin. We have our pelvic fins, which are paired, and our anal fin back here. So remember that your pelvic fins are going to come before, are going to be a little more anterior than your anal fin, okay? Because pelvic fin comes before anal fin. And then your pectoral fins are the ones on the side here. And you can picture your pectoral fins kind of being used like what you use your arms for when you're swimming. You can use them to hover. You can use them to swim backwards. You can use them to help you swim forward. So again, those are those pectoral fins. And you can picture like your pec muscles, where your pec muscles would be, okay? That will help you picture your pectoral fins. So is a pelvic fin ventral to the pectoral fin? So here's your pelvic fin and here's your pectoral fin. Which one is more ventral? Belly side, right? Okay. And so yes, the pelvic fin is going to be more ventral than the pectoral fin, which means that the pectoral fin is more dorsal, closer to the dorsal side back here than the pelvic fin is. Okay. How about anterior of the anal fin? So is the pelvic fin anterior of the anal fin? Remember, this is the anterior end of the fish. So let me just kind of write these as a reminder as to what your, so I'm going to put A for anterior, okay? P for posterior, okay? D for dorsal, and V for ventral, okay? So again, that can help you kind of remember. So, you know, if I say, is the pelvic fin anterior of the anal fin? Let's see, here's the pelvic fin. It's closer to the A, so it's anterior of the anal fin, okay? Or the anal fin is more posterior than the pelvic fins. Okay, so again, just a couple easy terms to help you orient yourself. This is really helpful, especially in lab, to be able to use these terms to orient yourself. And I will use those terms more again today when we do the, or sorry, not today, tomorrow when we do the dissection in lab. Okay, so some bony fish, again, anatomy. You're going to see the bony fish has a similar, I say, internal anatomy to your cartilaginous fish. We will talk about a few of the differences in terms of the intestines. So typically your bony fish will have these kind of long intestines, kind of long windy intestines. And then we're going to talk about the fact that the cartilaginous fish, especially your sharks, are going to have this spiral valve inside their intestine. So kind of interesting, but we'll see that more in lab. But just to give you kind of, again, an overview of some of the things that you're going to see in lab, you'll see the mouth is going to lead into a pharynx, just like us. And then that pharynx will then lead down into the stomach. And these guys typically will have a J-shaped stomach. We'll talk about why that is in lab. And the stomach, again, is going to be where they're going to do a lot of their digestion. And so you'll see in our perch, but they even have these this kind of special extra organ called the digestive ceca that will release digestive enzymes. They're going to look like these little finger-like structures. And they'll release digestive enzymes into the stomach. So their stomach is doing what our stomach is doing. It's primarily involved with grinding up that food and mixing that food with digestive enzymes to help break it down. Now, as that food moves into the intestines, okay, the intestines are going to continue to do some breakdown of the food, but their main purpose is going to be to do absorption of nutrients. So hopefully, again, as you break down that food and it continues to break down with the digestive enzymes as it makes its way into the intestines, then you have the food broken down into its basic components where it can be absorbed again through the intestinal wall into the bloodstream. Okay, so that's kind of the main function of that. And then, of course, then they can go ahead and expel that waste through the cloaca. Let's see, we'll talk about, again, we won't see the kidneys, but they do have a kidney, but they're really hard to see because they're tiny in the fish. The big thing that you're going to see when you dissect these fish is that big swim bladder. Okay, that's a big difference, that swim bladder there. And then you're going to see their liver is quite a bit smaller here, so they don't have that massive oily liver. Again, they're going to have a heart just like your cartilaginous fish that is two-chambered, and we'll talk about how that two-chambered heart works here. And then, of course, they have gills here, so you're going to see that they've got the gills along on either side of the pharynx. You'll see those gills. And the gills, of course, their purpose is to do gas exchange. Okay, so fish shapes. We're going to see different fish shapes in our fish depending upon, again, what habitat they're in and what their feeding niche is. So you can see a marlin, something like this is, again, going to be kind of a rover, what we call a rover predator. So it's going to be actively hunting down its prey, and because of that, it's going to have this very fusiform body. And you'll see that we call that fusiform body, any body form that's torpedo shaped. We say fusiform. Okay, so fusiform means torpedo shaped. And that just helps, again, to make the body of the fish more streamlined and to decrease drag. Now, a butterfly fish has a very different body form because, again, it's living in a different type of habitat with a very different feeding niche. And so butterfly fish are what we call laterally compressed. So it looks like you took the right side and the left side and you took your hands and you squeezed the fish and flattened it that way from the right and left side. So a butterfly fish, again, is laterally compressed because it wants this big surface area to, as it turns its body, to use that big surface area to make these big quick turns to be able to defend its territory. So this big surface area acts kind of like a paddle to be able to allow it to make these really quick, sharp turns so it can defend its feeding territory. And butterfly fish will do that, again, not ferociously, but adamantly. They'll do this quite adamantly because, again, their feeding territory is, their survival. So they're going to typically have these areas of coral where they'll pick off a few polyps here, but they keep that coral alive so that they continually can feed on that coral. So again, they are going to be quite territorial. So any type of fish, just as a heads up, that has a laterally compressed body is likely to be more of a territorial fish. Now, a flounder has what we call a depressed body form. Okay, and so this body form is going to be flattened like a pancake because, again, this fish, now it's had its eyes so in a flounder, right, the eyes are actually going to migrate to one side, either the left or the right side. So that's what makes it, again, a left-eyed or right-eyed flounder. And so in this case, again, if the pelvic fins are here, this is the anal fin, this is the dorsal fin, this makes this a right-eyed flounder. And again, they have this depressed body form. We call it depressiform because they're typically hanging out on the bottom. So again, different body form because of a different habitat. Anglerfish, again, are typically have what we call this globiform body form, and that's again because they're primarily sitting on the bottom. They want to look like a rock, so they have this kind of globular body that allows them to look like a rock, and they typically have these little spiny extensions and things coming off to look like algae, like they're growing algae on a rock, and oftentimes speckled, and they oftentimes, again, have that cryptic coloration that allows them to blend in with the rocks. They have this very large mouth, and so then, again, an unsuspecting poor little fish swims right by it. They open this large mouth, and it creates this suction and sucks the fish right in. So that's their kind of sit and wait feeding strategy. So they have a body form to match that feeding niche. And then, of course, eels are going to have this really elongated body form. We call it anguilliform, but you don't need to know that. And so this really elongated body form allows them to be able to move between crevices. Okay, so let's talk a little bit more about form and function. Again, fish that have these what we call fusiform or torpedo-shaped bodies are typically what we call rover predators. And they need this torpedo-shaped body to reduce drag so that way then they can actively hunt down their prey. So they're actively, again, chasing down their prey. So they typically have fins that are spaced out along the body for better maneuverability. Here you can see the first and second dorsal fin are spaced out. They even have finlets here in between, again, that second dorsal fin and the caudal fin, as well as the anal fin and the caudal fin. So those finlets help. Again, and also, and the finlets, as you can picture, actually help with stability as that fish moves quite rapidly through the water. And again, those paired pelvic fins and anal fin are spaced out. So that's more typical of what you're going to see in your rover predators that, again, are actively going after their prey. So you can see that in these fish here. And this is actually a tuna. So your tuna fish are actually in the mackerel family, as you can see here, because they have those finlets in between the dorsal and the anal fin and the caudal fin here. And then here you can see this fish is in the jack family because it has, again, that dorsal fin that just tapers down to the caudal peduncle. Here the anal fin just tapers down to the caudal peduncle. And then you just have a sailfish here. Again, those are all fish that have this very torpedo-shaped body, and they'll have these spaced out fins you can see here to really help with maneuverability because they are all rover predators. Okay. We have these fun surface-oriented fishes, and again, they often have these mouths that are oriented upward so they can capture prey that are primarily going to be at the surface. And so your flying fish are one of those fish, or we also call them flying gurnards. And you can see they have these really large pectoral fins to allow them to jump out of the water temporarily, and also it allows them to again, really that lift to bring themselves up towards the surface. So I have a quick video for you on, and let me see, I'm not 100% certain. I should warn you, this video may get cropped out because I do believe, I can't remember if this is a BBC video, sorry, and of course it has a long ad because of that. But I believe this may be, oh yeah, it's a live video, so let me see. It's Discovery BBC.
It's a dangerous place, whether it's a river, a lake, or an ocean.
So I'm not sure if it'll let me keep
Most
this.
dangerous of all is the open sea, because when predators strike here,
kind of fast forward so you can see these flying gurnards.
for most fish, that is.
So these are those flying gurnards, flying fish.
'Free of the water, flying fish soar on long fins adapted for airborne escape. but escaping predators is only half the battle. They also have to make sure there's a next generation. These flying fish are searching for something that will make this possible. A bit of flotsam. They're in luck. A palm frond. Perfect. A tiny raft adrift in a huge ocean. And, like a raft, it offers sanctuary, not for the flying fish, but for their eggs. The first females to arrive lay their eggs on the raft. Then the males swim by and release their sperm. Soon, thousands join in. Countless strands of eggs are laid and fertilized.
Open your eyes.
Okay,
to
so
the wonder
again, just a
of
quick kind of
life.
video on
Sunday.
the flying gurnards. So just to keep it interesting for you. I try to put a few of those videos in here. Oh gosh, it's going to open. Sorry, my email here. So, and that's my challenge with this. I need to reconfigure my computer, so I apologize there. Okay, so let's talk now about your depressed body form. So this is, again, typical of your bottom fish. So your bottom-dwelling fish, or your demersal fish, we call them demersal because they're bottom-dwelling, are, again, going to be a little variable in shape. Flounders and some sharks, or things even like sculpins here. Sculpin is a bony fish that feeds on invertebrates in the bottom. Again, a demersal fish. Are going to have these kind of, again, flattened body forms to help them sit on the bottom. So again, flounders are really flattened like a pancake. Again, there are some sharks that are really flattened, again, because they are bottom dwelling to be close to the bottom. Sculpins here typically have these modified pelvic fins here that allow them to actually adhere to the bottom. So look at these large pelvic fins. Pectoral fins. And then they do have modified pelvic fins as well. You can't see that in this guy, but they'll have these large pectoral fins that will help them kind of hold on to the bottom. But then they'll sometimes have these modified pelvic fins as well that are almost suction cup-like. And those pelvic fins, again, you can't see because the pelvic fins are on the underside here, but those pelvic fins will act kind of like a suction cup to help them suction to the bottom. So here again are your flounders. And so here you have a left-eyed flounder. So again, this is going to be the dorsal fin. These are going to be the pelvic fins. It's hard to see, but they're right up here. And then this is going to be the anal fin. So here the eyes have migrated to the left side, and you can see there's the mouth. So it is a left-eyed flounder. This is a left-eyed flounder. And then here you're going to see this again. The two eyes, have migrated here. Here's the mouth. Sorry, I should have put a right-eyed flounder in here, but this is another left-eyed flounder. You can see here is the pelvic fin right here, and one of them, and then there's one on the other side. So there's the paired pelvic fins right there. Here's the anal fin, and here's the dorsal fin. So this is another left-eyed flounder. So these are both left-eyed flounders. And you can see they typically will have this cryptic coloration where they blend. And this is actually a picture. This is one I think I took from the textbook. But this one I actually took in Puerto Penasco. And you can see it has this coloration to match the sand and the little white flecks from broken seashells in the sand. So again, pretty cool how their coloration really matches, again, the sediments. Form and function again, we have what are called these deep-bodied fish that are laterally compressed, okay? So they're, when we say laterally compressed, it just means it's like you took the right side of the body, put a hand on it, and the left side of the body, and then squished it in, right? Okay? And this makes them excellent at maneuvering. However, again, everything's give and take, right? So they give up this torpedo-shaped body for the maneuverability, but then now they're these slower swimmers because of it, okay? So they're excellent at maneuvering and defending their territories, but they can, again, swim and make these sharp turns really rapidly, but they're slow, again, swimming, so if they're being hunted down by a shark, it's, they're an easy prey because it is hard for them to swim fast. So these are butterfly fish. Again, just as a reminder for your fish family quiz, you can tell that they're a butterfly fish with this concave forehead. Again, an elongated snout with a small mouth at the tip of it for feeding on like individual coral polyps. They typically have, again, these stripes through the eyes and then these false eye spots as well towards the tail end. So again, and then they have this kind of very disc-shaped body. And then again, another very territorial fish because it is a deep-bodied fish. So again, this is going to be your damselfish, which it typically is not in distress and does the distressing. Again, and they have this kind of laterally compressed, laterally flattened body form that you'll see. Again, another form fits function is this eel-like body form, and again is well adapted to kind of moving in crevices, such as you're going to see in your moray eels here. So you've got some moray eels here. Here's some conger eels, just as a kind of reminder of some of the differences. You've got your moray eel here. You won't be able to see this very well, but again, the moray eel has this dorsal fin that starts kind of farther up on the head, and then they have no true pectoral fins. And then your garden eels, which you can't see their fins very well, but your garden eels are actually an example of a conger eel, although I'll give you a better conger eel for the fish quiz so you can see more of the structures. Now, I cannot talk about moray eels and not talk about the fact that they just recently discovered that moray eels are like straight out of the movie Alien. And that's because they found that they have this second pair of jaws, these internal jaws. And they call them these raptorial jaws inside its pharynx in here. And we call them pharyngeal jaws because they're inside the pharynx. You can see here's the pharyngeal jaw in blue here. And so what ends up happening is this pharyngeal jaw will be used to kind of lurch forward and grab the prey after the eel typically, initially bites onto a fish. And then it just draws it back in down the throat so it can swallow it. So really kind of interesting. So it reminds me of, Alien with these pharyngeal jaws. So if you want to know more about these pharyngeal jaws, just because they're pretty cool, I have a link here for you, and you can read all about these pharyngeal jaws. And they think that the moray eels ended up evolving these pharyngeal jaws because they typically, again, are found in these crevices and tight spaces. And so when they go to feed on a fish, they can't open their mouth very widely to just suck in the prey. So they really open their mouth wide enough to get the fish in, but it doesn't really suck the fish in, so they use these pharyngeal jaws to pull the fish in so they can swallow it. So they think that that's kind of an adaptation, speaking of feeding niches here and habitats, they think that's an adaptation to, again, living in these small crevices and these small spaces. So again, kind of interesting. All right, so fins of a fish. We're going to talk about how they use their fins to swim. So again, most fish will kind of swim with this side-to-side motion, and that has to do with how they're going to be moving their caudal fin. I'll show you this in some animation in from a research lab where they actually looked at the mechanics of a fish swimming. And since bony fish, again, have these swim bladders, the pectoral fins aren't needed to provide lift. So now the pectoral fins can be used for maneuverability, which is pretty nice for your bony fish. So now they can use these pectoral fins to hover, okay? And they can use their pectoral fins to swim backwards. Two things that cartilaginous fish cannot do, okay? So hint, hint. That's a big point, right? It's something that bony fish can do that cartilaginous fish can't do. Make sure you know that. The dorsal and anal fins are typically used more as rudders to kind of steer and provide some stability. And so that's why they can retract them, pull them up and down. And then the parapelvic fins are really used to help the fish turn, balance, and then really put on the brakes. So you'll see like in fish that, like your salmon that have to swim upstream, they have really big parapelvic fins to help kind of balance them as they're swimming against that turbulent water. And then also, again, they can use them to put on the brakes. I have a couple animations here. I'll go back to showing you some animations in a future slide where I talk about caudal fins. That has a really cool one of how the fish use this side-to-side body motion. So again, just really quick differences in bony fish versus cartilaginous fish. Remember that bony fish are going to have this cool organ called a swim bladder. And that swim bladder, again, is going to help them provide lift. It'll provide the buoyancy for them. And they're going to use Boyle's law, you'll see, with this swim bladder to be able to adjust where they are neutrally buoyant. And so I'll show you this video in just a minute. So bony fish use a swim bladder, where again, cartilaginous fish to provide lift have to have these big oily livers inside. They have these stiff triangular wing-like pectoral fins to provide lift, as well as that heterocircle tail, which is going to force the head up every time they swim. So again, some major differences here. And then this is just showing these muscles called the myomeres. These are really these, really pronounced muscles. And that's because the caudal fin is actually going to be where the fish is going to get most of its propulsion. So it's going to have these really large muscles that are going to be used to control the caudal fin and move that caudal fin back and forth. And this is primarily when we eat fish, what we're really going for. We want this muscle tissue, right? If we're feeding on fish. If we are eating fish, feeding on, sounds like we're pets. But anyway, so just to kind of give you an idea of how they're going to use that caudal fin. We'll talk more about that. So I'm going to let you watch this in terms of a quick video on how these swim bladders work. And they use, again, Boyle's law to move up and down within the water column. So hold on here for 1 second. Let's get through that.
Okay, so today I'm using this balloon to simulate the swim bladder of a fish. And I've also got some weights attached to it. So these are sort of simulating the weight of the fish that normally would drag it down. And in a fish's swim bladder, it's filled with gas so that it would keep it neutrally buoyant. And so what we're going to do is place it inside of this canister and slowly increase the pressure. Ready? So we're going to screw this on, and we seal it up so that we can increase the pressure. Make sure it's nice and tight. Got everything all connected now. So I'm going to flip on the pump, shut our bleed valve, and you can see water going in right now. Because there's not a whole lot of pressure on the gas inside that balloon, it takes up a lot of volume. It's basically displacing water. And by doing that, it increases its buoyancy. So the pressure in here is starting to decrease the volume enough inside that balloon that we can see it sink. Okay, so we're at about 50 psi right now, which, according to the Coca-Cola company, is how much pressure is inside of a can of Coca-Cola in a room temperature room. It is equivalent to diving down about 110 feet under the ocean. 15 psi is atmospheric pressure at sea level. And you can see that the balloon has shrunk quite a bit. When it went in, it was about the size of a tennis ball. Now it's about the size of a ping pong ball or so. And if I continue to increase the pressure, it will get even smaller. So the gas inside the balloon is being pressurized. So what Boyle's law tells us is that as you increase the pressure on a gas, the volume decreases. And also as you decrease the pressure, the volume of the gas increases. And that's because the atoms or the molecules inside the gases have space between them. And as you increase the pressure, those atoms and molecules get closer together. And so they occupy less volume or less space. Still the same number of molecules in there, but what happened was the molecules get closer because of greater pressure. And so the volume decreases. So now what I'm going to do is release all the pressure that's inside of here, and the volume of the gas is going to increase, and you can see it floats right back up to the surface.
And so again, you know, a fish would simulate that, right? As the fish uses its caudal fin to propel itself up, okay, then it's going to be more buoyant. As the fish it swims down, okay, it will swim down to a point where, again, there's enough pressure compressing the air in the swim bladder that it's now neutrally buoyant at that depth. So again, the fish use Boyle's law and along with the air in the swim bladder, they don't know they're using, of course, they don't know they're using Boyle's law. So I don't know if that cut me off, but so the fish doesn't know it's using Boyle's law, but intuitively it is, as, you know, it swims down. If it uses its caudal fin to propel itself down, you know, pushing against that buoyancy in the swim bladder until it hits a point where the air in that swim bladder is compressed enough that it's now neutrally buoyant at that depth. So kind of interesting. Now this was just for fun. Because there is a marine biologist that was owned a pet store, and you can see he actually created an artificial swim bladder for a goldfish that had a swim bladder issue. So let me pull this up.
Until a crafty man at a local aquarium shop stepped in. Eyewitness News reporter Alicia Nieves talked to the inventor of the goldfish wheelchair.
A few weekends ago, a woman walked into Aquarium Designs in San Antonio with fish she couldn't care for anymore. One by one, the fish were adopted, except for this one.
Unfortunately, there was one little goldfish that just couldn't seem to keep himself upright. He was stuck on the bottom, upside down.
The fish has a permanent bladder disorder. It can't float like the rest.
He was just so tough, and he wouldn't give up, and I knew I had to do something for him.
So Derek adopted the goldfish. He has a background in marine biology, so he tried different diets for the pet, but no luck.
These type of goldfish, you know, they've been bred, you know, a little more than some of the other goldfish, and so just like dogs and things that have some of the fancier breeding, you can get a little more health issues that can arise with fish like this.
Then Derek had another idea.
Fairly cool.
If the goldfish can't float by itself, why not engineer a device that floats for the fish?
I just started, you know, messing with some things and tinkering with different items and troubleshooting, and came up with the wheelchair that, you know, became famous, the goldfish wheelchair.
He used scraps from around the store.
Bits of airline tubing and airline tubing connectors. The base actually had plant weights that are wrapped around some zip ties and things to actually hold it down. And then the top is like some styrofoam pieces.
A friend posted a picture of the wheelchair on Twitter. In just days, it went viral.
He went from being, you know, upset, stuck on the bottom, to be swimming around and zipping around. His tail got going super fast. I mean, it's as happy as I've seen him since we've had him in our possession.
Well, the original device was...
It was just a fun video on how someone created an artificial swim bladder for a fish that had a swim bladder issue. Kind of interesting. I got to keep it interesting for you here. Only so many ways I can make fish bladders, you know, swim bladders interesting for you. So artificial swim bladders kind of spices it up, right? Just spice up the lecture a little. So, okay, so form and swimming. So the type of the kind of form that these different fish are going to use for swimming is kind of a combination of three different modes. So There's what we call the acceleration specialist, which has a body form and fin structures that are really designed for fast bursts of speed, okay? And so that's something like your barracuda. And then you have your cruising specialists, which are really streamlined, and their fins, again, are more evolved for swimming kind of long distances with little drag and as little effort as possible. And then we have your maneuvering specialists, which really have these laterally compressed, deep bodies to be able to help them maneuver, basically to be able to kind of cut back and forth rapidly. And so when we talk about all of the different fish, oftentimes they're kind of a combination of these three different types of body forms that are adapted for these different types of swimming. And I'll show you the difference between kind of the cruising and the acceleration, because we'll talk about, I'm going to show you a little short video on how some fish have adapted for like these short bursts of speed. So again, swimming usually involves this kind of undulation of the whole entire body. And so you'll see they'll undulate their body back and forth really to help them propel themselves with that caudal fin. And that's why they have these really strong muscles here called the myomeres that are really used to help their body undulate and really move that caudal fin back and forth. So these are kind of the components of force that they use when they're swimming, and that, so you can see why you don't necessarily have to understand all the mechanics of it, but I just wanted you to see why fish undulate their body. So they undulate their body really to produce thrust forward. So that's why they undulate their body back and forth. And it's really that caudal fin that's going to provide most of the thrust forward. So this is kind of a cool little animation here from a lab where they looked at kind of the mechanics of continuous swimming versus intermittent swimming, like these short bursts. So you can kind of watch this here. There's no sound to this, so you can see, you'll see these intermittent bursts, and then they kind of coast, where this is just kind of that steady coasting, steady swimming, coasting, and then you have these intermittent here. In this fish here, you have these intermittent bursts. So this is kind of cool. They looked at kind of some 3D modeling and flow simulation. And I'll just kind of let you watch this. This is just kind of interesting to see how they looked at water movements and how you'll see, again, the difference between your steady swimmers and your burst and coast swimmers. So this is cool. They looked at like the wake pattern in the water behind the fish. And so you can see steady swimming produces this kind of rhythmic wake pattern. And then you'll see more of a bursting wake pattern in these burst and coast swimmers. So watch this burst now. So kind of interesting to see, again, these different strategies and how the different fish will swim. So again, they look at some different surface pressures and things like that. So it gets into more of the mechanics than we are really going to get into. But you guys are welcome to kind of watch some of this. It's just interesting to see. I'm sorry, I need to turn off my notifications. You can see how they, again, are using those caudal fins and kind of how the water moves over the fish, which was really interesting. So again, just kind of for fun, to look at the mechanics of fish swimming. Okay, so let's get into some of the heart of this lecture, which is going to be the circulatory system, and then we're going to look at the respiratory system. So the circulatory system of fish is going to consist of veins that carry deoxygenated blood from the body tissues to the gills. So just like us, they're going to have veins that carry deoxygenated blood. The only difference is that it's carrying that deoxygenated blood from the body tissues to gills versus in us, it's carrying it to lungs, right? For what we use for gas exchange. Another big difference is that fish only have a 2-chambered heart, and we actually have a 4-chambered heart. So that 2-chambered heart is going to pump blood out to the gills for oxygen, and then on that single pump, it's going to go all the way from the gills out to the body tissues, dump the oxygen, pick up carbon dioxide, and then come back to the gills, all in one pump, okay? I'll talk about this in just a minute. And then the arteries are going to carry oxygenated blood to the rest of the body. So let's look at how it does this. And so really quick, let's also talk about why fish have a 2-chambered heart and why we have a 4-chambered heart. So again, we have a much higher oxygen demand, right, than fish do. Much higher energy needs, you know, especially being endothermic organisms that maintain a constant body temperature. We have a much higher oxygen demand to do that. And we have a much higher need for oxygen in terms of we have a lot more metabolic needs. So we need, for us, a 4-chambered heart that's going to pump, you know, our blood is going to pump deoxygenated blood to the lungs, and then it's going to come back to the heart. Okay, that oxygenated blood is going to come back to the heart after it picks up oxygen in the lungs and get a second pump before it's pumped all the way out to our body tissues, and then comes back. Okay, so we come to our heart goes, again, blood comes to the heart, gets a pump to go to the lungs, comes back to the heart, gets a second pump before it goes all the way out to our body tissues and back. And the reason we need to do that is, again, our blood needs to move more rapidly because we need that oxygen. Our tissues need that oxygen fast, okay? Because of our higher oxygen demand. Now, fish have a lower oxygen demand, and because of that, they can rely on one pump. So what happens is, again, blood in the tissues, as it, again, gives up its oxygen to the tissues, and becomes deoxygenated, it's going to get carried back to the heart, okay, where it comes in one atrium and then one ventricle. That's why it's just 2 chambers, one atrium, one ventricle. And so it'll come in, the atrium will collect the blood, pump it to the ventricle, and then the ventricle will give it one big pump. On this one single pump, that deoxygenated blood is going to travel all the way to the gills, pick up oxygen, then come out and travel all the way to the tissues here, where it's going to dump its oxygen, okay, pick up carbon dioxide, and then come back to the heart, all on that single pump, okay? And so again, fish can do that because they have a lower oxygen demand than we do. We could never get by with a 2-chambered heart because simply the blood would pump too slow to oxygenate our tissues fast enough, okay, for their needs. So that's why fish can get away with a 2-chambered heart. So how do fish obtain oxygen? Fish obtain oxygen by passing, again, just the water over their gills, right? And their gills are going to be doing the gas exchange. Their gills are not only going to pull oxygen out of the water, but they're also going to give up carbon dioxide. Okay, and the gills are going to lie in the pharynx, which is the front part of the gut. There are gills on each side that are going to share a common gill chamber. So oftentimes you have, 4 sets of gill arches in each gill chamber, and they're sharing one single gill chamber. And then there's an opening, okay, that's covered by the operculum. That's the gill, the operculum is the cover to the gill chamber. So it's very simple. The mouth is going to open and the operculum is going to close. That's going to suck water in to the mouth and pharynx, okay? And that pharynx is going to kind of relax and expand. That's going to allow water to suck in. Then the mouth is going to close, the opercula is going to open up, and the pharynx is going to contract, pushing that water out across the gills and then out the opercula. Okay, so it'll continually pump water in that way. So water, again, is going to flow over these folds in the gills called gill lamellae. And then oxygen will actually diffuse into the gills, and carbon dioxide will diffuse out into the water. So we'll talk about how they do this, but fish do this kind of cool thing called countercurrent exchange. And what happens is the blood flow is going to be in the opposite direction of the water flow to maximize their ability to pull in oxygen. And I'll show you how this works in just a minute. This is the same principle as for kind of heat conservation that we'll talk about in dolphins later on. So let's talk about this. This is just talking about the simple mechanism of irrigating, again, the gills. So first, to suck water in, they're going to open their mouth, close up those opercula, relax the pharynx, and suck water in. That's inhalation. Then in exhalation, they're going to close their mouth, open up those opercula, and contract the pharynx here, pushing water in across those gills and out the opercula. Okay. Now, when you look at gills, you notice that gills are really feathery. Remember what we talked about in the very beginning of the semester when we dissected the crab and we looked at the feathery gills of the crab? Remember we talked about that things that are typically feathery are that way because it's a way to increase and maximize your surface area. So if you think about it here, okay, I'm just going to kind of show you in terms of, oops, I need my pen here. I'm just going to show you kind of a quick drawing here. So if, we had just a flat gill like this, right? This is all the surface area that we would have. But instead, what happens is your gills have these folds called lamella, okay? And so here on each of these are called gill filaments. So let me kind of go backwards here and we're going to talk about maximizing surface area. So here for a single gill arch, you're going to have a gill filament coming off of this side and a gill filament coming off of this side, okay? So these are the gill filaments. Here's a gill filament and here's a gill filament. All of this is one gill filament. All of this is one gill filament. And then within these gill filaments, you're going to have these individual lamella. And, or lamellae is the plural. And what these lamella are simply these folds to maximize surface area. So watch this. Now we're going to put gill lamella on here. And look at how we can maximize our surface area. Okay. So now, instead of just doing gas exchange across this flat surface, the gill lamella here increase our surface area. So now we've got this surface area here, and it really drastically increases our surface area as the water is going to move through here, because now we can do gas exchange all along this surface instead of just this shorter surface here. So can you see how those gill lamella, those gill folds, you can think of the gill lamella just as folds. And these gill folds are really going to increase our surface area, okay? So again, pretty cool structure, and pretty cool way in which your form is going to fit its function. Okay. So again, each gill arch is going to have these two gill filaments coming off with all of these gill lamella. Okay. So here again, you can see this kind of shows you kind of a better depiction of what the gills look like. So let's look. Here is the gill chamber, and many of your bony fish are going to have what's called 4 gill arches. 1, 2, 3, 4. Okay, 4 gill arches on each side. Okay, now if we were to pull out just one of these gill arches and cut out a section of it, this is what it would look like. So here's your gill arch. And on the inside of this gill arch is what we call the gill rakers. You see those little spikes here? And they help to trap food and particles so that they don't clog up the gills as water moves in through the mouth and across these gills, okay? So as the water moves, again, sediments and things will trap in those gill rakers, and then the water will move in and across these gill filaments, okay? And then it'll be drugged this way. So you'll see inside these gill filaments, okay, here's the gill filaments, are going to be these, again, arteries and veins that are going to run through them, okay? And that's because, again, here we're picking up oxygen, right? So that's why when you look at gills, they're going to be red. They're going to be really brightly colored red. And the reason is that, again, they have blood vessels in them to do gas exchange. So that's their main purpose. So they're filled with blood vessels inside of them. And again, those veins are going to be carrying the deoxygenated blood to then pick up oxygen, okay? And then carry that oxygen back to the rest of the body, okay? So again, here's the gill filaments, and then you can see the folds here called the gill lamellae. These are the folds that kind of maximize our surface area, increase our surface area. So now this is cool. This gets into what we call this counter current exchange. And what it does is it's really smart. It maximizes your diffusion gradient. So what happens is as that water, again, that new fresh water that was just brought in the mouth, okay, comes in and moves here, it's going to have the highest amount of oxygen, okay? So the water is going to be like this with lots of oxygen. The red dots are supposed to be oxygen, okay? And so here's the oxygenated blood that's moving back, right? It's picked up some oxygen, it's moving back, okay? And so it's still going to have a little bit of a concentration gradient, and now the water is still going to have a higher concentration of oxygen than even the oxygenated blood from here, okay? And so what's going to happen is you'll continually bring in more oxygen into that blood. Now, as oxygen diffuses into the blood, right, the oxygen concentration is going to get lower, right? So as, again, some of the oxygen diffuses into the blood, our oxygen content gets lower. But what's cool is then now as it moves through here, right, it's going to hit the vein as it moves across this gill filament. And now the vein is deoxygenated. And so we're going to still have a higher concentration of oxygen. Even though it already gave up some of its, again, oxygen, it's still going to have a higher concentration of oxygen than the deoxygenated blood. So you'll still get, again, oxygen diffusion there as well. So it's just a cool way. We call this counter current exchange. And it's a cool way that fish use to maximize, or I should say that they've evolved. They don't know that they're doing this. This is, again, something that evolved over time. But it's this cool way to maximize their oxygen absorption and use that diffusion gradient. So again, this is called counter current exchange. Pretty cool. And so I'll show you an animation of this. So again, just as a reminder, bony fish have these 4 pairs of gills. Again, they're going to be bright red in coloration because they have blood vessels running through those gills, inside those gill filaments. Each of those 4 pairs, I say 4 pairs because there's 4 gill arches on one side and 4 gill arches on the other side. Each of those gill arches is going to contain 2 rows of gill filaments, okay? And then the lamellae are going to be these folds on the gill filaments that are going to increase the surface area. And they contain, again, lots of capillaries. So, again, diffusion of oxygen from seawater into the blood, it is going to use this countercurrent exchange. So water is going to flow across the lamella in the opposite direction to the flow of blood. That's that countercurrent exchange to make sure that you maximize your diffusion of oxygen, okay? And the concentration of oxygen is always going to be higher in the water than it is in the blood as it moves across via that countercurrent exchange, just like we talked about. So with countercurrent exchange, again, without that countercurrent exchange, there would be less oxygen absorption. So again, this really maximizes oxygen absorption in these fish. So I'm going to show you real quick this animation on respiration in fish, because I know it's kind of difficult to be able to, really difficult to kind of be able to visualize respiration in fish. Animation.
Fish are aquatic animals. Water contains oxygen dissolved in it. Depending on temperature and other factors, the level of dissolved oxygen in water is about 4 to 15 milligrams per liter. Fish extract this dissolved oxygen from the surrounding water by a process called aquatic respiration. Fish have specialized structures called gills to carry out exchange of gases with water. The gills are located on either side of the head of fish, behind the mouth cavity. In bony fish, the gills are covered by a hard, bony flap called the operculum. It is also known as gill cover. In cartilaginous fish, the operculum is lacking, and the individual openings to gills are called gill slits. During the process of exchange of gases, a fish first gulps water through its mouth. At the time of the intake of water, the mouth opens while the operculum remains closed. The fish then closes its mouth and opens the operculum to force the water out through the gills. The color of the gills is dark red or brown red because of the presence of a large number of blood capillaries in it. These capillaries come in close contact with the current of water crossing the gills. Blood absorbs the dissolved oxygen from the water by diffusion. The unique structure of the gills ensures that they absorb most of the oxygen dissolved in the water that is passing between them. Simultaneously, carbon dioxide from the blood diffuses out of the gill capillaries into the water. The water containing carbon dioxide is expelled through the open operculum. Besides being the respiratory organs, the gills also play an important role in maintaining the balance of salts in fish. Fishes like bluegill, seahorse, lionfish, Atlantic cod possess the hard cover of operculum. The best example of a fish having no operculum is shark. All the species of shark lack operculum and have only.
So they did a quick, I thought it was a nice job of kind of, other than they don't really talk about countercurrent exchange, but they do a good job of kind of summarizing kind of the whole process, and they give you kind of the big picture. So that was kind of the fine, but they do a pretty good job there. Okay. So what is hemoglobin? Hemoglobin, again, is a protein that we have, and the heme protein within the hemoglobin protein is red in color, and it's, again, contained within red blood cells. So hemoglobin is within our blood cells, it's within the blood cells of fish, and it's what's going to attract to oxygen. It's what's going to carry our oxygen within our blood. So it has a really high affinity, a really high love of oxygen. And it'll carry that oxygen from the gills to the tissues and carbon dioxide from the body to the gills. So again, it's kind of a misnomer that hemoglobin only carries oxygen. It does also carry carbon dioxide as well. And it'll carry that carbon dioxide from the tissues to the gills to get rid of that carbon dioxide, right, that you just saw in the animation at the level of the gills. Fish feeding. Bony fish, again, their feeding strategies are very diverse, and they have kind of two mechanisms in the water column that they're going to use. They use this kind of suction, where they rapidly open their mouth cavity, suck water in the prey into their mouth. Or, I mean, these are the two main fish strategies, but then you also have something like an eel with this extrapharyngeal jaw. So again, there are other feeding strategies besides these. These are kind of the two main ones, just as a heads up. And then you have things like ram feeding, where again, you have kind of a larger, more predatory fish that's moving forward, opening their jaws and encountering their prey directly. Many fish will chew on their prey by means of teeth, but remember that those teeth will be specialized for their different feeding strategies. So again, you may have more serrated teeth or teeth that are meant to puncture to grab onto their prey in your predatory fish, but that are again going to be feeding on other fish, or you can have some specialized crushing teeth, like you'll see in your pufferfish and sculpins, that are going to feed more on invertebrates, or going to feed on, again, kelp and algae and things like that. Some species, again, are suspension feeders, and they're going to trap zooplankton, phytoplankton, and again, organic matter. And they'll trap this plankton in what's called their gill rakers. Again, you're going to see this in lab. And we just talked about those gill rakers. And then they'll actually basically use the food that's on their gill rakers, and that's what they're going to feed on. So the gill rakers are going to trap the food, and then they'll kind of use their tongue to push off that food from the gill rakers back into the pharynx. So this is just kind of some different, again, different structures. This was actually from a sculpin, and these guys are shell crushers. So you can see here's their teeth that they would use to, crush into something like a snail shell. And then sometimes they just eat the invertebrates whole. So you can see these are bivalves in the fish gut of a sculpin. So kind of interesting. And then again, we like I just talked about, we do have suspension feeders as well. So this is an invertebrate feeder. Again, adaptations to deal with crushing into shells and exoskeletons. And then we have our suspension feeders, again, like your anchovies, or, even a basking shark, which is a cartilaginous fish. But again, you can see they're going to have these gill rakers that are going to trap again, plankton as they bring that water in and across the gills. And then they'll use their tongue just basically to lick off that food and push that plankton back into their pharynx. The feeding shape of their mouth is obviously going to be adapted as well for their different feeding strategies. So barracuda is pretty easy to see that, again, their mouth structure and this big protruding lower jaw is really meant for tearing chunks of prey off. And then here you can see in your butterfly fish here, you're going to have, these really kind of elongated snouts that are meant for feeding on very small prey. Again, individual coral polyps on your coral. And then here you can also have a slip mouth that's also, again, adapted for feeding on, your really small prey as well. And then here, so just two different strategies. Again, butterfly fish are feeding on really tiny prey. And then parrotfish is going to have a beak-like mouth. So again, the teeth are fused to form a beak so that they can graze on algae and coral. And then a herring is going to have a really large mouth, just like your anchovies, because they're filter feeders. So that way then they can open up that large mouth to bring in lots of water as they swim. We already kind of talked about digestion. I'm sorry that I did it earlier and not here. So I talked about, again, food going, again, from the mouth into the pharynx and esophagus, just like us. And then, again, eventually into the stomach. So here would be the stomach. And then, again, that's where a lot of your digestion is occurring, the breakdown of food. And some of them even have these pyloric ceca. So you're going to see all this in lab this week. So I'm kind of going through this fast. And then the food will go through the intestines, again, where you're going to have, again, a lot of absorption of the nutrients in the intestines. So that's why they're really long, because it maximizes your surface area. And then the liver will also help to secrete some digestive enzymes to break down fats. So just like our liver, their liver produces bile that will help them break down fats. And then that undigested material is just going to exit, again, the cloaca or anus. So again, some of your bony fish actually have 2 openings. Now your cartilaginous fish only have 1. They have what's called a cloaca, but your bony fish can have an anus and then a urogenital opening. So the anus would expel, again, your more solid wastes, and then your urogenital opening in some of your bony fish is a separate opening that would, again, release your urinary waste, and it's also used, again, for reproduction. So you can see the gonad here is attached to that. So a little different structure in some of your bony fish, in that they can have these 2 openings, an anus and a urogenital opening. Again, your cartilaginous fish only have 1. They just have the one-stop shop cloaca that does everything. It does, again, expelling wastes, and it does, reproduction. It does it all. So again, this is kind of just some of the differences between your bony fish and your cartilaginous fish. You can see again, here is going to be your cloaca, opening of your cloaca, and then here again you have the 2 openings, the anus and the urogenital opening. We're going to talk more about this in lab, where again they have these really large livers that you're going to see, the really large oily livers, and then we'll talk about this cool spiral valve that you'll see inside the intestines in some of your cartilaginous fish to increase the surface area for absorption. Okay. Sensory perception, we've already talked about this. So obviously your bony fish are going to have this lateral line just like your cartilaginous fish do. And again, there's these little mechanoreceptors, because they mechanically are going to be moving this kind of little hair-like structure that sticks up as water moves it. So, and this is used in, spatial location, in perception of approaching stimuli, because it's detecting kind of water turbulence, water movement. So again, it's detecting vibrations and water movements. They can even detect sounds in the water, the vibrations of sounds that move through water. That's detected on their lateral line. And then their eyes, again, fish often have excellent vision. And then they even have these otoliths, which are very similar to what we have in our inner ears. There are these little calcareous ear stones, and they're going to rest on these sensory hairs. And so they're kind of suspended, and they come in contact with these little tiny hairs that they're suspended on. And as the fish moves, those little calcareous stones will move on those sensory hairs as they move from side to side. And that will give the fish kind of or its spatial orientation of up from down, okay? So kind of interesting on those otoliths, very similar to what we have in our inner ear. And this is just showing you, again, that lateral line that I know you guys have seen so many times with that little neuromast sensory organ. So the neuromast cells in this nice little sensory organ that will actually get pushed and moved with the vibrations or water movements. Real quick, what is the function of schooling? So we see that our bony fish are going to school, and we see, again, this is kind of a behavioral characteristic of some aggregations of fish. Many of them are typically silvery side species that are oftentimes a prey fish. And sometimes these schools will form what we call fish balls or bait balls if, again, there's predators that are feeding on them. And they do this to help confuse predators. So you'll see, though, that fish don't they don't only school when there's a predator involved. There are other reasons that they will school. So So one of them is that schooling can help to reduce drag a little bit. So it saves on, the energetic cost of swimming. So kind of almost like, again, when, migratory birds are flying in formation, fish will school for the same purpose. So here is just a school of fish to kind of show you what that looks like. And you can see these are different common schooling patterns. So you'll see this is typically what they look like when they're, traveling together. This is what they look like when they're like feeding, maybe they're feeding on a reef or they're feeding on plankton. You'll see them feeding with this strategy. Encirclement of a predator. So again, if there is a predator, they'll typically swim in this kind of circular-like pattern to help confuse the predator. And then they can also streamline to avoid a predator that's coming as well. So again, just some different schooling patterns that you might see. As a reminder, fish have to regulate their body temperature. So most of your fish, remember, are ectothermic. So they're temperature conformers, and they just have to conform to the surrounding temperature of the water around them. Tunas and their relatives, like sharks, I say tuna and their relatives, and then some sharks. So sharks aren't really what we consider relatives of tuna, but tuna and some of the other predatory fish like tuna, as well as sharks. So more of like your tunas, your mackerels, a lot of your jacks, those more predatory fish, as well as some of your sharks can reduce heat loss. And they can have slightly elevated body temperatures by using these really powerful muscles that they have, the myomeres that are propelling their caudal fin, and that generates a lot of heat. This elevated body temperature will kind of allow them to have a slightly higher metabolic rate, but again, their temperature overall is still going to vary. You know, it might be slightly higher, but it's still going to vary with the surrounding temperatures. Okay. So again, it just allows them to have these slightly elevated body temperatures. Salt regulation in fish, just as a reminder, I know we kind of talked about this, but remember that the blood of your fish is going to be about 14, this is for your bony fish, it's going to be about 14 parts per thousand, where the seawater is at about 35 parts per thousand. So what's going to happen here is, again, water is in, remember that wherever salt's in a higher concentration, water's in a lower concentration. They're the opposite. Wherever salt's in a lower concentration, water's in a higher concentration. So water's going to move down its concentration gradient. So bony fish are continually losing water due to osmosis, okay? So this is water lost by osmosis through their gills and through their skin directly. So what they're going to have to do, and this is, again, saltwater fish only. Remember, this is only saltwater fish. So what they're going to have to do is they're going to have to drink seawater, okay, and then excrete those salts. So they can excrete the chloride from salts through their gills. And so again, they're going to have to do this. They'll also gain water and salts by swallowing the seawater with their food as well. And then what they're going to do is their kidneys are going to produce a really small amount of urine that's going to be pretty concentrated, okay? So a really small amount of urine that will have some salts, but very little urine, okay? And then some of the salts are going to pass through the gut, and they'll basically, poop the salts out, okay? So they basically, their goal is to drink lots of seawater and then secrete the salts through their gills and then through their intestines, in their waste, as well as in their urine, and then produce small amounts of urine. So hold on to as much water as they can, because they're continually losing that water anyway. Now remember, this is going to be very different from your freshwater fish, because the freshwater fish are going to be doing just the opposite, okay? If you are having trouble with this, please come talk to me, and I will help explain this in more detail for you if you need more help. Now, just for fun, I wanted to show you that cartilaginous fish actually do the opposite. Cartilaginous fish are not going to osmoregulate as well as your bony fish are. So what they're going to do is they're just going to take their blood concentration and match it to the surrounding seawater. So, and they'll do this by concentrating urea in their blood, which is a solute. So they'll just concentrate urea in their blood to help kind of match the solute concentration of seawater, so that they're not continually losing water. So kind of interesting. So one of the last things that we're talking about is counter shading. So remember that many of your fish, both cartilaginous and bony fish, will use counter shading. We already talked about this in lab, but this is that idea that they're going to be dark on the dorsal side, light on the ventral side. So that way then when you're looking up at them, okay, like here, that light ventral side is just going to kind of blend in and even washes out with the sunlight. So I've lost fish this way when I'm like looking and trying to like take a picture of a fish and it, you know, goes above me and I go to take a picture. I'm like, oh, I can't see it. So you literally can actually lose fish this way. And then of course the reverse happens. So as you're looking down at the fish, right, it's going to have this dark backside as you're looking down into the dark depths of the ocean. They'll have this darker backside. And so we call that counter shading. And many of your epipelagic fish will have this counter shading. You can even see this kind of counter shading in some of your deep water fish, and they'll actually have ventral photophores. So they have these, again, light producing structures to kind of trick your potential predators into thinking that, you know, there's some light coming through. So again, kind of interesting. You see that counter shading even in some of your deep water fish. And this is only for Thanksgiving when I teach in the fall, but these are all turkey fish. They're all have a common name of turkey fish. So I say, what do all the fishes have in common? Sorry that this doesn't work for you. What do they have to be thankful for? That they are not turkeys. So again, just kind of fun information for you. Okay, so that's it for the bony fish lecture. I know that was a lot. Please let me know if you have any questions on it. I am more than happy to go through some of this information with you. I went through it quickly because I know a lot of it was kind of review, but let me know if you need more on some of these topics.